Hello, everyone. Today I'm going to be talking about evolutionary traps and how larval lake sturgeon may be vulnerable to falling into one. I'd like to recognize my co-author and advisor, Kim Scribner, for helping in developing this study. So the adult decision of when to mate is very important for fitness. Adults use environmental cues to coordinate timing of mating between individuals, and this also predicts the conditions present for those offspring to develop under. Temperature is commonly used as a cue, however, it's becoming unreliable with climate change. So if a cue becomes unreliable, that means that there can be uncoordinated mating efforts between adults where adults are trying to mate at different times, so they cannot get as many mates as they may otherwise, or it can result in an unpredictable environment with poor conditions for mating and offspring development. This is the situation I want to be talking about today. So an example of this is, let's think of a flower that blooms in the springtime. So if this flower blooms at around 60 degrees Fahrenheit every spring, this could be good because that warm weather predicts abundance of pollinators, and it's safe for this flower to thrive under. Under climate change with higher variation in temperature, after the temperature hits 60 degrees in the springtime, there might be something like a April snow shower that comes, and as a result, the pollinators become dormant, and that flower is damaged and therefore does not reproduce. This is an example of an evolutionary trap where the adapted response, in this case, blooming as soon as it starts to get warm in the springtime, is actually detrimental due to the change in the environment. Cold-blooded vertebrates are especially vulnerable to these types of evolutionary traps because a lot of their bodily functions are completely dependent on the environment, and this includes lake sturgeon, my study species that I'll be talking about today. So a little background of lake sturgeon. They're originally threatened fish. They live well over 100 years and can spawn dozens of times, with males spawning on average every other year and females spawning every 3 to 5 years. They migrate from lakes up into rivers to spawn every springtime. They use environmental cues to guide the spawning efforts, including temperature as it's increasing in the springtime, as well as lunar phase, and also stream discharge, how fast the water is moving. So their subsequent larvae, after hatch, disperse downstream with the river current at night, and during this time, they're vulnerable to predation by a diversity of riverine predators. So all sturgeon don't spawn at the same time. There are distinct spawning periods, early and late period, at very different temperatures. So the early spawning period happens when the water temperature is right around 10 degrees Celsius. And here's some pictures from last year during this period. Very cold, not too pleasant to work in. And later in the season, when the water is 18 degrees, when we see that second large run of sturgeon come and spawn, and the environment's much different here. Everything's green. We can be in t-shirts out there instead. And as you can imagine, these are very different environments for the eggs and larvae to develop under. So when it's colder out, more of the yolk sac energy can be put into somatic growth, so we get larger larvae from this period, whereas later in the year, more of that yolk sac energy has to go into respiration to keep up with the high metabolism, and therefore we get smaller sturgeon. So with these distinct spawning periods, we also get distinct dispersal periods when those sturgeon are drifting downstream. These early dispersing larvae, like I said, are larger on average than those later dispersing larvae. Here's a graph showing the lengths of several families from earlier in the year versus later in the year. Earlier in the year, we see an average size at hatch of around 13 degrees or so, whereas later in the year, closer to 7 millimeters. It's almost double the size early in the year versus late in the year. We also see differences in the density of sturgeon and other larvae drifting downstream. So sturgeon are often found drifting downstream at the same time as larval suckers and a diversity of other invertebrates. Earlier in the year, we find many more of all of these major taxa dispersing than later in the year. And on average, there's 4 times the biomass early than late here. Also, these early dispersing larvae are dispersed close to a new moon, whereas a later dispersing larva disperse closer to a new moon. And this is important because we know that from a previous study that the probability of finding sturgeon in the diet of a predator increases with the amount of lunar illumination. Remember I mentioned that they disperse at night, so that lunar illumination is very important for the predators to be able to see and find these larvae, these prey. So as temperature becomes an unreliable cue, adults may spawn at seemingly inappropriate times, which could cause a mismatch of these early and late environmental characteristics. And this mismatch may negatively impact fitness. So our objective in this study here was to determine whether larval sturgeon are vulnerable to falling into an evolutionary trap by answering the question, do these mismatched conditions decrease the survival of these larval sturgeon? So here's our experimental design. We set up these flow-through mesocosms. So water would come in here and flow downstream here. We had 2 rock bass separated here by some gates, and we released larval sturgeon, larval mayflies, and larval suckers from the upstream end, allowed them to flow downstream past the predators, similar to what would happen naturally during that nighttime dispersal period. And we captured all survivors downstream of the predators over the outflow in these nets. We varied 3 factors in these trials, either the size of sturgeon by raising them at either 10 degrees Celsius or 18, giving us much larger and smaller sturgeon respectively. We also changed the density. So we either released 32 of each of these taxa per hour versus 8 per hour, reflecting that 4 times difference in biomass from early and late in the year. And we also varied the light, either setting it to 0 lux, which is the conditions in a new moon, versus 0.2 conditions during a full moon. We did this with 8 families of sturgeon, 8 male-female combinations, and for a total of 64 trials. Then we analyzed it with a mixed model GLM, looking at survival of the lake sturgeon as a function of size, density, light, as well as the interactions between these, with family as a random effect. So here I'm going to show you some interaction plots. Here we have the proportion of surviving sturgeon, and here we have the light levels, so either dark new moon light levels or full moon light levels, and here we have the density split between high densities and low density trials. So early in the season, this is the combination of environmental factors we'd expect to find, where here's what we expect later in the season. Now, if these factors were mismatched, these high density populations dispersing under much lighter conditions would have a drastic decrease in survival, whereas there would only be a minor increase in survival among these low density groups. So overall, this mismatch between light and density decreases survival. Next up, we have the interaction between rearing temperature and density. Here again, we have the large sturgeon versus the small sturgeon, and split up again into the high density versus the low density. This is the combination of effects from early in the year versus later in the year, and really similar to the last graph I showed. A mismatch in conditions here would again decrease the survival of these high density fish, while not having much of an impact at all in the low density fish. Again, the mismatch between these rearing temperature and density factors decreases overall survival, providing evidence that they're susceptible to a evolutionary trap. And lastly, here we have the interaction between rearing temperature and the light conditions. Here we have large larvae, small larvae split up between the dark trials versus the lighter full moon trials. This is what the combination we expect early in the year versus the later in the year. And this time, if there's a mismatch in the environmental characteristics present, there really isn't much of a change in the survival of the sturgeon drifting under the darkest nights, but there's a pretty drastic increase in temperature in survival for these sturgeon that are drifting during the new moonlit conditions. So in this case, actually a mismatch increases the total survival of these larval lake sturgeon. In conclusion, larval lake sturgeon are susceptible to evolutionary traps. This is particularly important for the high densities of lake sturgeon to be dispersing during the new moon darker nights, and those larger colder sturgeon dispersing at high densities. It's important to remember that predators would be much more abundant in the wild than they were in my mesocosm experiments here. Here we had 2 predators over 24 feet, where in the wild they'd have to disperse over several kilometers and be exposed to hundreds of predators. Also, it's important to keep in mind that low recruitment would result in slower recovery for these threatened sturgeon populations, and these evolutionary traps could contribute to even lower recruitment. Also shows that larval abundance should be monitored, and we should determine whether these mismatch ecological conditions are occurring in nature, and we should be tracking how they're influencing recruitment. Lastly, I'd like to acknowledge Doug Larson and Lydia Wassing particularly for helping out in getting these experiments going and everything at our sturgeon rearing facility, and the huge crew of people we've had over the last few years up there have helped a lot in getting everything. Also, our funding is provided by Michigan DNR. I'd like to thank you.